Hi, everyone. Welcome to the Health First Infection Prevention and Control podcast. We've got another exciting topic to talk about today. It's exciting and very timely. On this podcast, we'll be talking about COVID-19 vaccines, what you should know. It's really timely given some of the late breaking news about vaccines that are being administered only in the past couple days. In this podcast, we'll be talking about the main vaccines available in the US and some common concerns. This discussion will be led by two leading experts in their respective fields, both representing the medical and dental fields, Dr. Scott Cohen and Dr. Fiona Collins. So I'll hand over the mic over to Dr. Collins for her perspective around the main vaccines in the US.
Thank you, Grace, and thank you everyone for joining us. So let's start by looking at the main types of vaccines that are available specifically in the US. These are either messenger RNA, or known as mRNA-based vaccines, or they involve protein-based technologies. None of the vaccines involved use live virus, and none of them use inactivated virus either. So there's no SARS-CoV-2 virus involved in any of them. I'll start with the two mRNA viruses and then hand over to Scott, who will address the other three vaccines currently involved in clinical trials. So as we look at the mRNA vaccines, I think at this point we're all aware that the two that are currently available are from Pfizer and Moderna, both very, very recently released. They are both mRNA-based, and they have a lipid nanoparticle coating. So they're encased in this coating. The Pfizer one has 4 lipids, doesn't really matter. The Moderna one has 3 lipids. So basically that is protecting the mRNA. So how do these work? Well, let's look first at the SARS-CoV-2 virus itself. We all know at this point that one of the coronavirus proteins is a spike protein on its exterior, and this spike protein binds to the receptors before fusing to our own cells and entering them. So that logically makes a focus on the spike protein as an antigen for which antibodies are created in our bodies, an attractive proposition for a virus. If we look more specifically now at the mRNA that is being used for the vaccines, it encodes the spike protein and it's encased in this lipid nanoparticle coating. When it's administered, it's actually the coating that fuses to the cell, after which the mRNA enters the cells in a similar fashion to SARS-CoV-2 itself. The mRNA uses amino acids and a couple of other chemicals in our bodies naturally to create the spike proteins. We're not creating the virus, only the protein. Our body then recognizes it as a foreign body, causing an immune response, the same as any other foreign body would cause an immune response. So as is typical for an immune response, it involves the release of macrophages that will then migrate to the area and destroy the cell. In the meantime, a signal is also sent to our B cells and our T cells as part of that immune response. The B cells produce the antibodies against the spike protein. The T cells really act as guards for us. They're watching out for the next time the antigen might be encountered. In other words, in this situation, the next time would be the spike protein on the coronavirus itself, conferring immunity to fight that coronavirus before it can replicate and cause disease. I'm now going to hand over to Scott, and he's going to talk about the technologies involved in the other vaccines.
Thanks, Fiona. Yeah, I get the easy one, thankfully. Most of what I'm going to talk about for the next minute or two, I can say what she said, because the end product of the mRNA vaccine, of course, is producing the spike protein particles, which then, promote an immune reaction, both likely an antibody or humoral reaction, as well as a cellular mediated one. But the reality is, when you're doing this type of vaccine that I'm talking about, which is an antigen or protein-based vaccine, you're using a vaccine that has been around for technology-wise many, many years. For example, the hepatitis B and flu vaccines use this same technology. Now, maybe the viruses aren't grown exactly the same way. We've all heard about chicken eggs and things like that. But the reality is what you're doing is you're taking specific proteins, antigens, you're mixing them up in a bowl, and you're injecting them in a certain way so they elicit a reaction such that the body produces a response. Now realize that those antigens are part of what is contained in that RNA virus. And what happens is then when you then see the virus, a real virus, weeks, months, or years later, then the body's immune reaction is re-engaged, and the body says, wow, I know that, I don't like it, I'm getting rid of it. Already has a reaction kind of stored in, B and T cells, i.e. humoral and cellular immunity. And then what happens is you get what's called somewhat of an amnestic response, i.e. In a more aggressive response than if it's the first time you're seeing that, which is why typically with a lot of viruses or other things, you get it once, you build an immunity, then the second time your body has enough memory cells of sorts to kill it off before it becomes, clinically evident or clinically significant. It's interesting when we look at the initial goals of the FDA, the goals for getting approval, and we're not talking formal approval, we're talking about what has actually happened with Pfizer and likely to happen with Moderna in the next day or two, and that's the emergency use authorization. The FDA's goals were 50% efficacy. And certainly I'm not sure whether it's 50% reduction in disease, 50% reduction in severe disease. I don't recall what the numbers were specifically, but we vastly beat that. And basically all of the vaccines under most circumstances are over 90% effective. And honestly, Anthony Fauci, said this, best, I think, and that is, it's just quite amazing that we're able to get these kind of results. And, Fiona will talk about safety in a moment, but I can tell folks that this technology has been worked on for many years. Now, the fact that we just implanted the coronavirus spike proteins into this technology is new, and that's what's happened over the past 9 to 10 months. But the technology has been worked on for many years now. So don't think it's quite as new as people say, or people are worried it is. It's something that's been studied, for quite some time. So with that said, common questions come up, like I'm going to pass it off to Fiona. Is it safe?
So really, so far as we've seen, the safety profile looks really good and really promising. We don't know long-term effects yet, if any, because obviously there aren't enough people who've been vaccinated for long enough. So it's still going to be followed through the remainder of the clinical trials, as well as obviously in individuals who are receiving the vaccine to prevent COVID-19. So if we look at local and systemic reactions, the local reactions that for both Moderna and Pfizer have basically been redness, swelling at the injection site. It's been a higher percentage of people who received the vaccine compared to those who received the placebo. But as you know yourself, when you've had inoculations, it's not unusual to have a little bit of pain, sometimes more than others, depending on sometimes who gave it, as well as some redness and swelling. So it's not unique to this at all. There were no serious adverse local events, so nobody was going to the hospital because they had redness and swelling and pain. It was not at that level that required intervention. The most frequent complaint was pain. I don't think that's any surprise either. So then if we look at the systemic effects, they were obviously also noted and researched, and the frequency was actually a little higher in younger people than older people, which personally I found to be a little surprising, but that's how it turned out. The majority were mild and moderate in severity. So it was things that we would probably have expected. Fatigue, headaches, muscle and joint pain in some cases, some nausea or vomiting, chills and fever. There were some reports of swollen lymph glands or lymphadenopathy that were it was described as being plausible that they were related to the vaccine, but not proven. There were 4 serious fevers, what we call grade 4 level in the Pfizer trial. However, 2 were in the vaccine group and 2 were in the placebo group. And there were no other systemic serious reactions requiring hospitalization or an emergency visit in the trial. Vomiting and diarrhea in that trial was similar between the vaccine and the placebo. Serious adverse events were extremely rare. There were 2 cases of anaphylaxis in the Pfizer trial, the cause is unknown, and there were no severe hypersensitivity reactions in the Moderna trial. In the Moderna trial, there were also one severe nausea and vomiting case and 2 facial swellings. They resolved or responded without any further complications. Now, if you think about the number of people that were tested, I don't recall the exact number for the Pfizer trial. It was a huge number. And in the Moderna, it was more than 30,000. So it's really overall a very good safety profile. And with that, I think Scott and I would like to do some myth busting as you described it, right?
Absolutely. Yeah, I think, there are certain side effects or worries out there that, we can, I guess, start off calling a myth, maybe are not a myth, have some basis in truth. Some are clearly myths and, are not even in the realm of having a serious discussion about. But the first one that I really liked is microchipping. So this is not a federal government attempt to microchip everyone in the population such that they can be tracked. Not at all. But there's another one. So what do you think, Fiona, about the antiplacental antibody issue? This is something that's come up.
Yeah, there was a suggestion by 2 researchers or former researchers that it causes female sterilization because it contains a spike protein that has something called synctin-1 in it, which is important for placental formation, therefore it's antiplacental. In fact, there's no basis for this whatsoever. The Pfizer vaccine, and it was about the Pfizer vaccine, this particular suggestion, does not even contain synctin-1. It contains messenger RNA or mRNA, as we talked earlier, and it trains the immune system to identify the real virus. It does not contain the substance that this suggestion is based on. So clearly a false concern. What about genetics? What have you heard about our genetics in relation to the, I think that's another of the myths that's going around.
Yeah, this is a good one. And, I had heard, and even on a webinar I gave yesterday for our local county health department, there were people that were floating around, is it true that, this vaccine, mostly they're talking about the messenger RNA vaccines from Moderna and Pfizer, but can they change your DNA, meaning change your genetic structure? And I think Fiona and I clearly agree, and the evidence is out there that is absolutely not the case. You're not going to change your DNA and morph into a different species or something. So I would say that's no. What about the other one? This is a really interesting one, Fiona, that kind of just perked my ears up, although it turns out has probably a rational explanation. And that is, what about the incidence of Bell's palsy?
Right, so but Bell's palsy, we couldn't really put under Mythbusters. We did, but it isn't really, you can't say it's a myth at this point. So there were a few Bell's palsy cases. Now, again, the first thing to remember is how many people were in the tests, both in the control and the vaccine, and the control vaccine group, and the vaccine group and the placebo, which was the placebo control. Mm-hmm. So as we look at that, there were 4 vaccine recipients and none in the placebo arm of the trial who did develop Bell's palsy. However, when they looked at that, the level, the incidence of Bell's palsy in this group was no different to the base incidence in the general population. So it was no different to the general population. For the Moderna one, there were 3 in the vaccine group and 1 in the placebo group. The cause was determined to be unknown, and there were predisposing factors. So they are going to watch on this, but in the first trial, it was the same level as the general population. And in the second, the 4 individuals who did develop Bell's palsy had predisposing factors. One of the other ones that we've heard about was the PEG, the polyethylene glycol. What are your thoughts on that, Scott?
Yeah, so polyethylene glycol is a chemical that's used in medicine, different than ethylene glycol, which is used in automobiles and is quite toxic to the kidneys in higher doses. But polyethylene glycol can have a small but distinct profile for causing allergic reactions. But we think that the likelihood of it being the triggering factor because of how much is in the vaccines is pretty low. And I don't know, Fiona, if you have the specific numbers. I don't really have them here.
I think for one of them, it was something like 0.05 milligrams. The other aspect is when you think about PEG, it's been used in the US for many purposes since the 1930s. It's in foods for various reasons. It's in shaving creams and gels. It's in other types of gels. It's in deodorants, cosmetics, fragrances, hair products, and other products as well. So it's not an unknown ingredient by any means.
No, not at all. The last one we had under our Mythbusters category is narcolepsy, that these vaccines cause narcolepsy. Now, can any vaccines, as first get them and they cause fatigue and malaise, cause fatigue? Of course, but not narcolepsy per se.
Right, and the other interesting thing is
okay.
This was in relation to one vaccine, and they've looked, CDC did a retrospective on over 1.5 million people looking at H1N1 and seasonal flu, and there was no increased risk for narcolepsy whatsoever.
Excellent. Okay, so we're gonna go through just a few more things here. Some questions, these are kind of related to some questions that have come up from our local population, but things like how long will it last? We're not 100% sure. We do believe initially you get a humoral or antibody immunity that's measurable, and that's why people have tested antibodies for COVID exposures. But then there's probably a much longer term cellular mediated immunity where you can actually maintain immunity for a bit longer time. We're thinking though, probably we'll need an every year booster until this virus is eradicated. And if this virus is eradicated, so very similar to influenza. We don't think it has the antigenic migration that you see in the flu. So it's not like we'll have to change the vaccine and hope and pray that it hits the right strain here and there, H1N1 or whatever. We really feel that it's fairly antigenically stable in that sense because of the proteins that we are targeting. Those proteins at least are stable. One other question that came up that's similarly related, and it's a really good question that we do not have an answer to, is, let's say, I'm a healthcare worker, I get vaccinated, but then I accidentally get exposed to disease. Not that I'm going to change my personal protective equipment regimen after I get vaccinated, I will not. But let's say I do get exposed. Can I still, even though I have no symptoms, and even though I've been vaccinated, can I transmit this disease to someone else? That's an as yet unknown. And I don't know, Fiona, if you had any thoughts on that.
No, I think we all have to take all the precautions we've been taking, both in professional lives, but also in our personal lives, and not go into stores without a mask on, for instance, once you've been vaccinated, because there are lots of other people behind you in the queue or the line who have not yet been vaccinated. And so we just don't know the answer to this any more than we know exactly how long it's going to last for.
Very true. So briefly, we're going to get into just some of the meat of how the vaccines are being delivered, distributed, and immunized, and then we'll kind of wrap up for you here. The delivery method and timeline for this is really a little bit up in the air. We know that FedEx and UPS have been intimately involved and have helped folks, like Pfizer, set up these pretty unique shipping and monitoring box devices. Really kind of cool. They've been doing it for months, so that now that when they're shipping, they know how to do it. And that's probably how initially they're going to do it. And we've seen initially Pfizer has done that and has gotten that out to local areas. Now, realize, of course, that there's going to be multiple avenues for getting this out there. And each of the manufacturers is using a different distributor, but there's local health departments, hospitals, pharmacies, and then, we don't know yet, but we might wind up using these federal points of distribution or PODs through health departments. That's all up in the air yet because of the focus right now on getting specific groups immunized. And I don't know, Fiona, if you want to kind of give an update on what it looks like the initial rollout's going to be too.
Yeah, based on what we know, the initial vaccinations will be going to essential healthcare workers, but in particular, the frontline workers who are exposed to COVID-19. And really, if you think about it, they're putting themselves at great risk, more risk than anybody else, and it's absolutely appropriate. You hear people want to skip the line occasionally, but this is totally appropriate. And after that, it will be other medical care and other healthcare workers, as well as high-risk individuals. But of course, it's depending on the state. So some states will prioritize some groups more than others. The over 65 is another group that will be prioritized. So what happens in places like prisons, some states will prioritize them because they're in a close living environment over, for instance, the general population. I don't know what you've heard. I heard that by May, June, if all goes well, that the people who want to be vaccinated will have been able to at least have their first dose by then, which would actually be incredible.
Yeah, I agree. It looks like that will be the case. We're hoping high-risk folks by February, March, at the latest April, and then everyone in the population, hopefully within a month or two after that, which honestly is just absolutely amazing that the public health system plus manufacturers and government have been able to bring this all together in record time.
Yeah, and in the meantime, as we go along towards that goal, we're going to get a lot more information from the remainder of the trials and a lot more information from how the distribution is going and how the vaccination rate is going as well, which will be important.
Absolutely. All right. So I guess with that said, we just want to thank you for coming today. Please, everyone get vaccinated and look forward. Obviously, we're going to do some other podcasts. We'll look forward to talking to you then.